A question was asked like this: talk about the silent points or silent features, right? So they could say silent points or silent features of the Watson and Crick model of double helical structure of DNA, right? So I saw the same question in another exam that we were saying that what you should just talk about the silent features of DNA. So they could ask it directly. They could ask it and include the name of this Watson and Crick, okay, because they're the ones that actually propose that double helical structure of DNA, right? Saying that DNA actually has two strands like this. So silent features, what does silent points or silent features mean? Silent points or features simply means some characteristics that are unique to this thing. That once you see it, you just know that this is DNA, right? So you can see that it is double helical. You can see that there are some turns. Some turns are big, some turns are small. So those are the things you actually talk about. So let's move into the lecture. So you have to, if they say you should talk about the Watson and Crick one, you have to actually acknowledge this year, right? It's nice, okay, to actually acknowledge the year. It makes the examiner relax and know that okay, you actually know what you're talking about. So the Watson and Crick model of DNA was published in 1953. It was a groundbreaking revelation about the structure of DNA. Okay, so here are some key points about it. So the Watson and Crick actually noted that the DNA has a double helix, like double strands. Like, see, this is a strands coming like this. These are another strands coming like this, right? Double helix, right? So the DNA is composed of two strands that form a double helix, resembling a twisted ladder. It's like a ladder they are climbing, but it's twisted. Okay. Then complementary base pairing. So these strands are held together by what? Hydrogen bonds between the pairs of nitrogenous base. So looking at this now is you can see between this, right? The blue one is guanine. So this is guanine, this is cytosine, right? So in between guanine and cytosine, you can see a black line. That's basically the hydrogen line joining guanine and cytosine and the rest, okay? So you find it that adenine will be paired to thymine and guanine will be paired to what? Cytosine. Okay, moving to the next one now. The DNA has some antiparallel what? Strands. So the two strands run in opposite directions, with one strand orientated from the 5' to 3' and the other strand oriented from 3' to 5'. Okay. Then it also has a sugar-phosphate backbone. Okay. So each strand has a backbone made up of what? Alternating sugars. So if we're talking about DNA now, it's basically what? Deoxyribose. Okay. We are talking about RNA now, that's when you're talking about ribose. Okay. So it has what? A backbone made up of this alternating sugar, that's the deoxyribose and phosphate groups. Okay. The replication. The model suggests a semi-conservative mechanism of replication. That's when you talk about the event of DNA replication. But I've already answered about two questions on event of DNA replication. All right. Now, this semi-conservative mechanism of replication, it simply means that what? Each strand serves as a template for a new complementary strand. You get? So that's it for the silent features or salient point of Watson and Crick model of double helical structure of DNA.